Maria Veldhuizen from Mersin University. Maria is going to be giving us a talk entitled "Too Salty, Your Amygdala-Thalamus Gain Network May Need to Loosen Up a Bit." Maria, thank you very much, and we'll look forward to your talk.
Thank you so much. And I would also like to thank, I guess, the Neuromatch algorithm, because I think the symposium is pretty well composed in terms of overlap between the different talks, as you will see, hopefully. So I'm going to talk a little bit about taste perception. Taste acts, or the function of taste is basically to be a gatekeeper of our internal milieu, and basically the perception of different taste qualities functions to help us identify harmful or neutral substances. For example, bitter and sour tastes are unpleasant and could be potentially signifying spoiled or poisonous foods. And then there's the aspect of intensity perception of tastes, which will indicate the quantity of substances. And so why is this important? Well, we think that these aspects of perception are related to food preferences, a diet, and risk for obesity and alcohol consumption. So I'm going to take this example that I think everybody will relate to. So if you go out to dinner, you will almost always find a salt shaker on the table. And that's because people's levels of salt perception just differ hugely. So for example, I may actually send the plate of food back to the kitchen because I think it's too salty to even eat. And another person may say, you know what, it's pretty strong, but it's all right. And another person may add like a whole bunch of extra salt when you're eating the same dish at the same table. I wanted to tell you, well, this is an interesting observation. I think that there are such huge individual differences, and an indication of why that may be came from taste illusions. So you probably know about all kinds of visual illusions. There are also taste illusions. One of those is called thermal taste. And what happens with thermal taste is that if you have pure water in your mouth and you warm the tongue, or you warm that water from 20 degrees to a couple of degrees higher, up to about 37, 40 degrees Celsius, what will happen is that intensity of the sweetness will go from the intensity of the taste will go from no sensation to between weak and moderate sweetness for about 30% of people. So again, there's no taste present at all in this stimulus. And what is also the case is that those 30% of people that perceive thermal taste also perceive in general other tastes as stronger than people that do not perceive thermal tastes. And it has also been observed that in general, if you perceive one taste as stronger than, so say, for example, sweet quality, then you're also more likely to perceive salty, sour, and bitter as stronger in intensity. So all of this suggests that there is some kind of central mechanism. We know already that genetics also play a role, but that there is about 40 to 60% variance that is unexplained, and that is thought to be environmental, but that can include central factors. And so each taste is a good predictor of sensitivity to other tastes, which, knowing that the different receptors in taste have different mechanisms, this indicates that it might be central. So our question was, what might be that central mechanism? And there are some indications for that in the literature from patients who have had their amygdala resectioned for epilepsy. And patients will perceive taste as much stronger compared to after surgery compared to before surgery, or compared to people who haven't had any, who have intact amygdalas. Which is kind of counterintuitive, because usually you would think that if you take away a brain region, this would lead to a weaker sensation. So our prediction here was that we will, like in those studies with thermal tasters, we would observe cross taste quality intensity correlations, such that when one person perceives one taste to be strong, they would also perceive the other taste qualities to be strong. And that it would be specifically connectivity between amygdala and earlier taste areas in the taste pathway. And we were focused on insula-thalamus in this case. And that connectivity would correlate with intensity, and that specifically that connectivity would be inhibitory, consistent with that observation that if you don't have an amygdala, your taste intensity perception is stronger. So in this study, we used 4 different taste qualities, so sweet, sour, salty, and bitter, and we presented the same intensity to all participants. And we collected data in 30 healthy weight participants. We first did a session, an event-related session, where we collected taste ratings, 8 ratings of intensity for each of the taste qualities. And then we ran a scan, an fMRI scan, where we presented the tastes in a block design, but people didn't perform any task, so passive sensation. And so this is what a trial looks like in the event-related design, where we collected the ratings. First, they get a taste stimulus, then they swallow, they make their intensity rating, they receive a tasteless rinse, and then they swallow again. While during the scan, we used a block design where we see multiple repeats of one and the same taste, and then at the end of a block there is a rinse. This is what our scan sessions look like, more or less. We have computer-controlled pumps that we use to control the delivery of tastes in the mouth. We have a special mouthpiece that sits comfortably between someone's lips and in a consistent manner drips tastes onto the tongue. So concerning the intensity ratings, here I graphed the intensity ratings for all participants for each of those taste qualities. And on the right side, you'll see the scale that we used with those labels of from barely detectable to very strong. The horizontal arrow bars actually indicate the variance from those 8 different ratings that a participant made, and then each little circle is 1 participant. And so what you can see is that we have a lot of variation. Some people perceive the tastes to be barely detectable or weak, and then some people very strong. And in general, we see more variance in the bitter taste, which is kind of a known thing because there's a lot of genetic variation in bitter tastes. But we also see that the different taste qualities are not on average perceived differently in intensity. And so, oh, I'm sorry, I skipped through that a little bit fast. We're going to actually use the average of the sucrose, the citric acid, and the sodium chloride, so sweet, sour, and salty, to get our intensity metric here, because quinine showed a little bit more variation and correlates less strongly with the other taste qualities. So you can see that here too, that in the left column, you'll see the correlations between sucrose and the other tastants, where it's not significant for corrected for multiple comparisons if you look at the correlation with quinine, but it's very significant with sodium chloride and citric acid. And this agrees very much with other studies that used larger groups of participants that showed those cross-quality correlations. So when we look at the main effective taste in a voxel-wise analysis, we see sort of our usual suspects of responses in the insula and in the pre and post central gyrus. I plotted here for the averages to all tastes and all tasteless blocks. And as you can see, almost all participants show a difference in the positive direction for taste blocks. So we have now addressed two of our assumptions, that each taste is a good predictor of sensitivity to other tastes, although quinine is not great, and that gustatory cortex robustly responds to the taste stimuli. So now we can go on to our prediction about connectivity. So where is in the brain is response to taste versus tasteless correlated with that intensity metric? So when we do a whole brain correlation, we observe a correlation, a positive correlation in the left amygdala. And we also had some unpredicted areas. I didn't include those in this presentation. And then if we ask the question, where is connectivity correlated with that intensity metric? We used a PPI analysis, psychophysiological whole brain correlation, and we used seed regions that showed a main response to taste versus tasteless, or regions that showed a correlation with that sensitivity metric. And we basically only observed significant correlations when we used that amygdala as a seed region, and we observed bilateral significant connectivity with the left medial dorsal thalamus and the pulvinar, or the ventral medial area that is specific to taste, but that's also so small that it's kind of hard to know whether it's really that falls within one voxel pretty much. So then we wanted to take those regions, those 4 thalamus regions and the amygdala, and use a DCM model. And I thank Dr. Matsurana for introducing the DCM models, because I didn't prepare a slide for that. And we specifically wanted to we used Bayesian pruning to find out which of those connections show posterior probability above 95, and whether those connections were inhibitory. And we observed that from the amygdala to all of those thalamus areas, that was the case, that there were inhibitory connections that correlated with sensitivity. And if you take that multivariate model, then you see a positive correlation with between the predicted intensity from that and the actual observed intensity. So concluding, that means that we identified a region of amygdala where response to a series of tastants is correlated with overall gustatory system responsiveness, that amygdala region drives inhibitory outputs to the gustatory and limbic nuclei of the thalamus, and that the dynamics and strength of these inhibitory connections predict individual variation in taste intensity ratings. And so this circuit acts to create gain in the gustatory system to regulate taste intensity perception. I would like to take a moment to thank Dr. Small and Dr. Green, who were involved in this study. And I would like to take questions now. And don't do this in your lab. I can do this in my lab, but nobody else probably can.
Thank you very much. Thank you, Maria. That was fascinating. What an interesting talk. We are taking questions.